This is the story of aerobic respiration. Aerobic respiration takes place inside a mitochondrion. You can see that the mitochondrion has lots of infoldings called cristae. The first stage of aerobic respiration is glycolysis. In glycolysis, you break down glucose and you start the reaction using 2 ATPs. That releases the energy and you get left with 2 ADPs. You also produce reduced NAD and overall 4 ATPs. So in total, this set of reactions leads to the production of 2 ATPs and a reduced NAD, and 2 lots of pyruvate. The next is the link reaction, and that takes place in the mitochondrial matrix. So we transport the pyruvate across the mitochondrial membranes and into the mitochondrial matrix, which is just like cytoplasm, but inside a mitochondria, so it's called matrix. The pyruvate then combines with coenzyme A and forms acetyl coenzyme A. With this, you lose a carbon dioxide. So this compound is only 2 carbons big, whereas pyruvate had 3 carbons. Next, after the link reaction, you go into the Krebs cycle. During the formation of acetyl-CoA, you also release another reduced NAD. The acetate loses its coenzyme A and is released into the Krebs cycle. The acetate is only 2 carbons, but when it combines with oxaloacetate that's already in the mitochondrial matrix, you get a 6 carbon citrate. The 6 carbon citrate very quickly loses a carbon dioxide which diffuses out of the mitochondria, and this produces a 5 carbon compound. This 5 carbon compound also loses a carbon dioxide, so you make a 4 carbon compound. Whilst that is being occurred, you also reduce another NAD. All these reduced NADs we're going to come back to in a moment. From there, the 4 carbon undergoes a number of changes, and this results in the production of ATP, a reduced FAD, and another reduced NAD. Finally coming back to the 4 carbon compound oxaloacetate. Don't worry about knowing the names of these intermediates, just know that they're 4 carbon compounds, and between the 5 carbon and oxaloacetate you get 2 reduced NADs, 1 reduced FAD, and 1 ATP for each turn in the Krebs cycle. All of those reduced NADs and reduced FADs come in handy. They're reduced because they all carry H plus and an electron. This is very useful and is used in the mitochondrial cristae. So here we have a section of the mitochondrial cristae membrane, and here it is blown up. So this is the inner membrane, this is the outer membrane, and this is the intermembrane space. We have protein carriers and an ATP synthase enzyme. And basically all that's happening is the excited electron bounces along the electron carriers, releasing energy. That is enough energy to drive the movement of the H pluses, which were released from the reduced NAD and the reduced FAD, and get them pumped into this intermembrane space. The fact that you're accumulating so many H plus ions sets up a diffusion gradient, and hydrogen ions want to diffuse back out into the mitochondrial matrix. However, the only way that this can happen is if the hydrogen moves through the ATP synthase coupled protein channel and enzyme. So basically the only way out for hydrogen ions is if it passes through this protein. As soon as it does that, it drives the enzyme that replaces ADP with ATP. So by adding the phosphate using the energy from the movement of the hydrogen, you get ATP. So for every reduced NAD, you get 3 ATPs, and for every reduced FAD, you get 2 ATPs. The hydrogen will then combine back with the electrons and oxygen and water will be produced. And so now you can see where the carbon dioxide and the water are produced in aerobic respiration.